Hiya. Okay, so this is section 2 of key area 4 on ante and postnatal screening, and in this video we're going to be covering genetic counselling. Now this is the inheritance patterns, and we're also going to be looking at postnatal screening, which is a tiny bit on PKU. Now, important thing to start off with, the inheritance patterns will definitely be in your exam. I have never yet seen an exam paper that does not have stuff about genetic counselling, so mainly it's about an inheritance pattern. So we'll be looking for something on autosomal dominant or recessive, sex-linked or incomplete dominance questions. So that's a very, very important key area. Unfortunately, we're about to explain why we can't do it terribly effectively over a single video with you.
Okay, so in terms of interpreting genetic test results, so obviously certain people can be carrying a specific genetic disorder, certain people can show a certain genetic disorder, certain people might not have it at all. But knowing and being able to look at these patterns allow us to understand more about who would then pass this thing on to their offspring. Because maybe you carry it and don't show it, that doesn't mean you don't have a chance of passing it on just because you don't show it yourself. And so this process of genetic counselling lets us look at people's genetics and really interpret, well, who is going to pass this on and who is incapable of passing this on to potential offspring.
You go through this one as well.
Okay, so in terms of stuff that you learnt at Nat 5, this is a topic that most people absolutely loved or absolutely hated. There was no kind of in between. You learnt about phenotype, so the idea that that is the physical expression of a gene, so what your physical appearance actually looks like as a result of this gene that you have. You learnt that the combination of alleles in a person would be their genotype, so if they are got two dominant letters, a dominant and a recessive, or two recessive letters, when we talked about it being homozygous or heterozygous. You talked about the idea that the genotype is very reliant on what the phenotype is. Again, heterozygous or homozygous will depend on that. And you also learned about the idea that genes can be dominant or recessive. So if you had two capital letters, that represented it was dominant, which meant it was kind of that overpowering trait. If it had two little letters, it was recessive. If you had a heterozygous mix, so a capital and a small letter, it meant it was heterozygous, but then the dominant trait would show over the recessive trait because it was that kind of more overpowering gene, so to speak.
Right, now with genetic counselling, the idea is that genetic tests can be very expensive and not all carriers of genetic diseases can be detected using these tests. So what genetic counselling is useful for is it looks at family trees and phenotypes to determine the genotypes of all family members. It's essentially a person whose job it is to sit down with a National 5 inheritance question and go, okay, so we've got a brown-haired person here and a brown-haired person here. You have a 25% chance of producing a brown-haired child. Essentially, that is their whole job. Okay, so it helps to inform prospective parents about the risk of, instead of passing on brown hair, it's genetic diseases, because they tend to follow the same rules as characteristics like tongue rolling or earlobe type. Is you get dominant genetic diseases and you get recessive genetic diseases, and you also get something that's a little bit in between that we'll cover in a bit.
So in terms of the patterns of inheritance, there are certain patterns that you need to be able to identify, you need to be able to understand, and most importantly, you need to be able to answer family tree questions using the idea of this pattern. And they are autosomal dominant, autosomal recessive, sex-linked, and incomplete dominance. So 4 different things. And the key for these is whenever you get a question about these, the question will say which one it is. And if you know what the thing is, it is going to make it so much easier for you to understand. If you do not know what each of these mean, you have not got a hope in being able to do the question, because they are very, very specific things and they do different patterns.
The favourite one tends to be sex-linked because there's a little quirk about writing genotypes on there.
And it's the most mean one.
It's the most mean one, aside from maybe incomplete dominance. But it does tend to be, the favourite one tends to be sex-linked. Now, something that is going to help this make sense is to understand what on earth the word autosomal means. The word autosomal means, if it's an autosomal gene or an autosomal genetic mutation, what this means is you find that gene or that genetic mutation on chromosomes numbers 1 to 22. You do not find that genetic mutation in the X or the Y chromosomes. It's not on the sex chromosomes. Now, what this means is a knock-on impact is equal numbers of males and females can be affected by it because it has nothing to do with sex. Okay, so nothing to do with X or Y chromosomes, nothing to do with sex, which means it does not discriminate between males getting it or females getting it, because it's just on the chromosomes that every human has in common, numbers 1 to 22. Now, you don't have to name those numbers 1 to 22. What we're trying to get across is just the idea that autosomal has nothing to do with sex, so equal males and females are affected. Now, as we're going through these exercises, or these examples, we will be referring to genetic diseases. If we're talking about an autosomal recessive gene, we mean an autosomal recessive genetic mutation. We are talking about the mutation in all of our examples.
So in terms of autosomal dominant, there are certain features that you need to be able to identify. And again, if you see a family tree and you can see these 4 features, that means it's showing autosomal dominant. So the first thing being the idea that there is an affected person in every generation. Because it is a dominant trait, if it is passed on, whether you have 1 copy or 2 copies, so whether you're heterozygous or homozygous, if you have it, it will show. It will be there so that you will be affected. So there will be an affected person in every generation. So if you get to the point that 2 heterozygous have a child and then they pass on the recessive, that means that that disease is then gone. It can't happen anymore because the dominant gene has basically been
It's
evolved
not physically
out.
possible for it to skip a generation. It can't skip a generation. So we say, as it exists, it's in every generation.
Yeah. Also, the idea that it affects males just as much as females. Now, that doesn't mean you're going to have 7 males and 7 females, but it means if you've got maybe 8 males, you might have 6 females, you might have 10 females. It will be similar, but not necessarily the exact same, which is one thing that does confuse people, is it's not the exact same. Like 2 and 3 is still considered similar amounts of them. If it was 40 compared to 2, yeah, it's probably not going to be this because that's not similar. I've already said fact sufferers can be homozygous dominant or heterozygous because it's a dominant trait, and for someone to be healthy, they have to be homozygous recessive. So they have to have 2 copies of the recessive allele because the idea is it is a dominant thing. So if it's dominant, then people who are homozygous recessive don't have it. A common example of this is Huntington's disease.
You don't need to know the examples, you just really need to know the features of them.
Yeah, so this is an example of a family tree that shows autosomal dominant. So you can see from the key, and when you do get a question like this, you will always have a key showing like who is what. So what is a male, what's a female, who's affected, who's unaffected. So you can see from this, if it is a black square or a black circle, it is showing an affected either male or female. If it is a white square or white circle, it is an unaffected male and an unaffected female. So you'll notice there is 4 affected males, there's 3 affected females, so pretty much equal numbers, so that's a good sign that it's autosomal dominant. There is an affected person in each line of the family tree, so the very top line, second, third and fourth, so basically going through your generations from your parental and your F1 and your F2 and so on, it's showing that there is people who have inherited it in every single line, which shows that it is, again, most likely to be autosomal dominant. And actually the best way when you get these family trees to work out who has things is if you can find out someone who is homozygous recessive and work back from them, because they are the person you can guarantee is going to be 2 copies of the small letter. They are generally the easiest person to work back from to find out who else is what, because you can work out their parents based on that, because their parents must be heterozygous, and then from so on you can work back for these, because quite often you'll get a question in this where they'll say, okay, this is person F, here's someone 3 lines above them, what is this person's genetics? And you have to use the facts that you know to work back to that.
If we take the bottom row as an example, we can say that every single black coloured in person, so every single affected person on that bottom row is heterozygous. They're going to have 1 copy of the affected gene and 1 copy of the unaffected gene. And the reason why you know this is because of, respectively, the father on the first load of children and the mother on the second. Those unaffected people had to pass on the unaffected gene. Okay, we can also assume that their parents are also heterozygous, so both the sets of parents are also heterozygous because they have managed to produce unaffected children. They have passed on 1 recessive copy of the gene, the other parents carried on a recessive copy of the gene. So things to watch out for in these questions, just very, very quickly, is look for the text above the question. The text above the question should tell you, in 80% of cases, it tells you this is autosomal dominant or this is autosomal recessive disease. Okay, so look for that. If it doesn't, that's where it's going to ask you in the questions underneath what type of inheritance pattern is shown here. So you need to look for the features of equal males and females every generation. So it's dominant every generation, equal males and females, autosomal. So that's quite easy feature.
And it's worth knowing that if it doesn't give you a letter, I would choose the letter to do it. That was about Huntington's. Use big H and little h, so a capital H and a little h, and write all the ones that you know on it, because it's going to help. If you can write all the ones that you know, everything that comes off of it is going to be easier if you can already be like, well, I know what 8 of them are.
However, note of caution on that, if they do provide a letter, you have to use that letter.
Yes.
If you decide to change the letter, if they say using the letter M and you decide to make it an F, you will lose all your marks, even if you get the capitals and the non-capitals correct.
And don't choose a stupid letter that looks the same as a capital and a small letter. Like, don't use a C or an O.
Don't use those are bad. Okay, autosomal recessive. Now, again, this just makes sense. It's a recessive condition, so you need to have both of the little letters in order to show it. What this means is it can skip a generation. The word autosomal means equal males and females affected. So sufferers have to be homozygous recessive, so they have to have 2 bad copies of the disease gene in order to have it. Carriers are heterozygous. So what a carrier is that's a person who's carrying the disease but isn't affected by it. So they are perfectly healthy, but they are carrying the gene for the disease. So depending on their offspring and their partners, they might have affected offspring. Unaffected are homozygous dominant. So people who don't even carry the gene, they are homozygous dominant. An example of this is cystic fibrosis. You don't need to know this for an exam, but to give you a statistic, about 1 in 20 people carry the gene for cystic fibrosis. Okay, so what this means is they have 1 copy of the healthy gene and they have 1 copy of the disease gene, and that means they're carriers of cystic fibrosis. Patterns of inheritance that it show, here we can see it's skipping a generation. We've got about equal males and females affected, 2 to 1, that's about equal. Okay, so that's the pattern that we're seeing there. And the idea is, again, we're seeing it skipping a generation. Now, again, everybody who is unaffected, we do not know whether they are homozygous or heterozygous because they are perfectly healthy. They might be carriers, they might be no carriers at all. However, everybody who is colored in black, everyone who's affected, they must have 2 little letters representing their genes. Okay, they're for the 2 mutations of the autosomal recessive disease. Okay, so remember, we're talking about all these examples. We're talking about an autosomal dominant disease, autosomal recessive disease.
And again, remember, if a child is homozygous recessive, both parents must be heterozygous if they are showing that they are unaffected, because they have to pass on 2 copies. They have to get a copy from each parent of that recessive gene, so each parent must be heterozygous if they are producing an affected child in this case.
Yep, so if we look at the bottom row, the parents of the bottom row, they must be heterozygous. Both of those parents have to be heterozygous.
So sex-linked, so this one pretty much does what it says in the tin. It is to do with your sex chromosomes, and specifically it is to do with the X chromosome of the sex chromosome. So obviously we know on your chromosomes 23, if you're female it's going to be XX, if it's male it's going to be XY. On the X of that is where sex-linked ones are linked to. Y doesn't matter. There's nothing to do with that. It's just there. It means you're a male, but it doesn't have anything particularly to do with this. And we'll explain exactly how it works in a wee minute. But the idea is that it's all to do with the X chromosome of the sex chromosomes there.
Let's try and think sex-linked, X-linked. Now, sex-linked features, more males are affected than females. And the reason for this is males have 1 chance to get it right, whereas females have 2 chances to get it right. So males basically can end up with, if they end up with 1 bad gene, they're going to have that condition. Whereas females, if they end up with just 1 bad gene, we're going to assume the other one is healthy, and this is going to mean, because it's recessive, that they will be healthy overall. Now, there are various rules about X-linked or sex-linked conditions, and we're going to go through them. This is the one that they like the most in the exam papers, and again, we're going to cover extremely unsatisfactorily on this, because the best way to learn this is find examples of exam papers and just work your way through them. Get a teacher to confirm it and work through it with you, because it is hard, it is difficult, but once you've got the rules in your head, suddenly it makes sense, and then it just clicks and suddenly you're away. So affected fathers cannot pass their condition to their son. So if you've got an ill father, he cannot pass on the disease gene to his son, because the father passes on the Y chromosome to their sons, not the X chromosome. Okay, so they pass on the Y chromosome, they cannot pass their condition on. Now, second rule is this weird one. Genotypes are written a bit differently. Now, we still use big letter, little letter, or big letter, but you have to attach it on to the chromosomes that person has. So for females, you write 2 X chromosomes, and then you attach each letter that you want to write to each X chromosome. So for example, let's say we've got, using the letter R, we've got a person who is homozygous recessive, you'd write X little r, X little r. And that just, again, we're saying little r, but we just attach it onto the X chromosome. Males are written, again, we write the X and the Y, but because the gene is not on the Y chromosome, we leave the Y chromosome blank. Nothing goes there. We don't put a 0, we don't put a little scribble, you put absolutely nothing next to the Y chromosome. Features of X-linked as well, or sex-linked, it can skip a generation. Unaffected males will have unaffected or carrier daughters because they pass on their unaffected X chromosome, and that depends on the mother. So the mother will choose whether they're a fully unaffected female or a carrier daughter. Okay. Now, example of this is hemophilia. What we see with hemophilia, it affects more males than females. It can skip a generation, and it's passed on by females. So female carriers have a tendency to pass it on to their sons. Is what
Another one that's quite nice is the colourblind one. Is that generally everyone knows a man or two that is colourblind. Nobody really knows girls that are colourblind because it is very much, it is passed on because it's sex-linked. It's males get it. Loads and loads of males are colourblind. It is so rare for a female to be colourblind just because of this. They've got 2 chances to make it correct, and chances are they don't get it.
And that's because, again, the colourblind gene or the colourblind mutation is found on the X chromosome. So sex-linked shows this kind of inheritance pattern. This is deeply unsatisfactory from a sort of point of view of trying to explain it to you, but we'll try. So what we've got up top is an affected male who would be X big R Y, let's say that for that, and they pass on their big R X gene to their daughters. Okay, so that's all they can do. So both daughters end up as carriers there in the circles. And then the carrier, if we take the one on the left as an example, the carrier daughter, to make that affected son, she had to pass on her X, I made this big dominant, R. I've made this dominant, annoyingly, X big R, they had to pass that on. Okay, so what we're looking at is carrier daughters pass it to their sons. We've got 3 red affected males. We have 0 affected females on this, so we're saying more males affected than females. It's skipping a generation in terms of its visibility. We do not count carriers as part of the affected generation because they don't actually have that condition. Okay,
so getting towards the end, this is the final pattern inheritance that we're going to talk about, and it's the idea of incomplete dominance. And actually, the best way to see this is in plants, annoyingly. It's the only time we're going to ever really talk about plants, but the idea is that sometimes you have alleles that are not necessarily more dominant or recessive than the other alleles. And it's really nicely shown in plants because the idea would be if you have a red plant and a white plant, they can make a pink plant. It's just like in art when you mix the colours. They just mix in quite nicely, and that is a showing that the red and the white are not necessarily dominant over each other. So to make up for that, they just mix, they merge, and then they make a nice in-between kind of colour. So for example, as it says here, so using the allele for red being a big R, but the allele for white is also a big W, because both of them are not necessarily dominant or recessive over each other. So we're going to give them both a capital letter just to show they are equally dominant, or there is incomplete dominance because one is not dominant over the other. So if we had a white flower, which is capital W, if we had a red flower, which is capital R, they would make a pink flower, which is capital R, capital W. So it shows a bit of both. And again, the flowers, it's just nice because it's just colours. You can see it. You can see very clearly this is how colours mix. This is obviously present in genetic diseases, but none of them are quite as easy to see as just here's 2 flowers becoming a middle colour flower. But this is called incomplete dominance. But this one tends to come up less. It is the sex-linked one that comes up the most.
Incomplete dominance tends to come up as a sentence answer question saying what's the name of the condition where neither are dominant or recessive, so it tends to be a sentence. Okay, so features of incomplete dominance is you get partially affected people. This has never happened before. Carriers have always been totally healthy before. Okay, but in this case, we've got a half a disease happening. So you can have a really severe form of the disease and you can have a half of the disease, which is a bit strange. But that's because, again, you have a healthy gene, you have an affected gene, you get a half healthy, half affected person. There are more carriers than sufferers. Now, that's just statistically it's more likely you're going to end up with a mix of the characteristics than two identical characteristics. Sufferers will be homozygous for the mutated gene. So they'll have disease as their genotype, okay? Unaffected people will be homozygous for the normal gene. So they will have healthy as their genotype. Carriers will be disease healthy as their genotype, okay? Males and females equally affected. We tend to not complicate matters by putting incomplete dominance on the sex chromosomes. An example is sickle cell anemia or sickle cell disease. Now, you can have a person who is perfectly healthy and they have no sickle cell anemia or disease. You have a person who is healthy one gene, sickle cell disease on the other, and they have something called sickle cell trait, which means effectively they are half affected by this disease. And you can have a person who is sickle cell disease for both of their genes. That person is very, very ill and they have full-blown sickle cell disease. Okay, so that's again, you don't need, it's not an examinable answer. I keep bringing up sickle cell disease and anemia because it's a nice mutation as a substitution and all that kind of jazz, but it's not one that you have to recite in an exam situation. So what they look like is you've got partially affected people, okay? Notice the statistics there. We've got more partially affected people than we do fully blown sick people. We've got just about the same number of partially affected people than healthy people, okay? And the idea is that partially affected people can again pass their condition on to make partially affected people. So if we look at individual 6 has passed on their sickle cell, one copy of their gene, to individual 5, on the bottom row of generation 3, okay? Whereas individual 5 in generation 2 has passed on their healthy gene to person 5, and so you end up with a partially affected person. If we look at example person 3 on generation 2 and person 3 on generation 3, person 3 has handed over one copy of their disease gene because they didn't have a choice in what they were going to hand over. Person 4 has handed over a healthy gene to person 3 on the bottom row, and that's created a carrier who's going to be half affected by the condition. A lot of these diseases, it's quite unusual to see people who've got the full-blown trait reproducing because full-blown diseases do tend to cause fertility issues or at least health issues that mean that the person can't really reproduce.
Or
so it's
die
quite unusual.
before you do, sadly.
Or you die before you do, yeah.
That's generally it.
Right, now that is us off that topic. That was a long one, sorry, 22 minutes that we've gone on for that one. We'll cover postnatal screening really, really quickly. So postnatal screening is after the baby is born. It's usually done using this technique. So the baby is born, they stab them in the heel and collect a sample of blood.
Okay, the final thing that we're going to talk about is the idea of PKU, and this is a common thing that is screened for in postnatal screening. And it stands for phenylketonuria. I don't think you have to know that, you just have to know it's PKU. So it is basically it's a metabolic disorder where you can't break down phenylalanine correctly, which is a type of amino acid, and basically it means that you have a really big issue with processing proteins, because phenylalanine is found in all proteins. So basically you have to avoid anything that has too much protein in it because it could result in brain damage and eventually death. Now, it is a really, really serious disease. People who have it really, really need to watch their diet. So this is why it's screened for so early. Like as soon as the baby's born, it's screened for because it could kill them within a day or two if they accidentally have too much breast milk because it contains a lot of protein. So really, really important this gets screened for immediately when you're born. I'm assuming all of us get screened for this as soon as we're born.
Now, if you think you're going to forget about PKU and you're going to forget it, the YouTube link will be in the sway. It is of a woman and she will say the word phenylalanine at least 16 times and you will never forget it for as long as you live. It's a brilliant video. It's a fantastic one. So I really highly recommend clicking that link if you can get onto the sway to do it. Now, for PKU metabolism, this is the way it works. Phenylalanine cannot be broken down, okay? Now, in non-PKU people, it's broken down to tyrosine, then other metabolic products. This is a common question that links back to metabolic pathways. They really like to fling in PKU after covering a metabolic pathway, something to do with protein breakdown. So, people with PKU cannot break down phenylalanine into tyrosine, and instead what happens is the phenylalanine starts to build up and is converted to other very toxic compounds which damage brain cells. So this is why it's so important to restrict protein intake in PKU babies, because you will cause irreversible brain damage to them if you allow them to take in too much protein. Even once or twice it can cause the damage. Okay, so this is what happens in sufferers of PKU. They build up toxic metabolic products because they cannot convert it to tyrosine and then to other harmless.
And again, this is just a picture of the kind of diet you'd be recommended if you had PKU. So obviously the ones on the left, your very high protein things. You will all have heard of things that are high protein. The things like your meat, your fish, your dairy, all these things. These are things, if you have PKU, you should not be having any of. Basically, if you have PKU, fruit and veg, sensible diet. Obviously, it's a sensible diet for us all. But if you have PKU, having things on the left will kill you.
Okay, now PKU is an autosomal recessive condition. So what this means is equal males and females are affected. Both parents have to be carriers to pass the condition on to offspring. So both parents must hand over their one recessive gene, second recessive gene, homozygous recessive person. It cannot be predicted by standard antenatal testing, so that the condition can come as a little bit of surprise to parents, and they do have to strictly enforce their child's diet in order to prevent serious mental disability in the future. That can be a challenge for quite a lot of parents, given that it's also in artificial sweetener. So you get a lot, if you look at the back of things like chewing gum packets, it says contains phenylalanine. That is for, or phenylalanine, that is for PKU people. That's why that's on there.
Even flavoured water, that's something which is a huge level in, which is not something you expect to be high in it.
It's because it's an artificial sweetener. Okay, now to summarise, so that was PKU, super nice and short, this is the most important thing out of this video, is these 4 inheritance patterns. So an autosomal dominant condition, every generation is affected, equal males and females, only one copy of the allele is needed to be affected.
Autosomal recessive, it will skip a generation. There's still equal numbers of males and females affected, but 2 copies of the alleles must be there for someone to be affected.
Okay, in sex-linked, your very basic generation, female carriers, male sufferers, and also remember that rule about writing the genotype. It's usually in an exam question because people forget to do it. It's a trick.
And incomplete dominance, carriers can be partially affected, so 2 copies of the allele result in fully affective. That's the one where they are both equally or unequally dominant.
Okay, so that is the end of ante and postnatal screening. It's quite a big one. Yep, sorry about this video, looking at 26 minutes.
Do the past paper questions. That is the best thing you can do for this. Is just practice, writing them down.
And get help from your teacher. We are aware that there's people watching these videos who are not taught by us. Welcome. You're more than welcome to access these videos and that sort of thing. But we really, really recommend the best way to learn these is practice it, get help from your teacher in working through the worked examples, and then it will click. It's like your times table. Suddenly, just one day, oh, I understand how to do this. And don't forget the rule about writing the genotypes for sex-linked. That's the main thing I can stress from this.
XX and XY.
Next key area, we are moving off fertility stuff into heart stuff. So the next key area is about arteries, capillaries, and veins. And the rest of this unit will be to do with heart health and general health matters to do with that.
Yeah, it's quite nice.
Yes.